Thank you so much, Abdel El Quaamari, for joining us from New York City. Just a quick bit about you. He completed his undergraduate and graduate school at the University of Nice, Sophia Antipolis in Nice, France, and he earned a bachelor's degree in biochemistry, a master's degree in life sciences and health, and a doctoral degree in aspects of molecular and cellular biology. He trained in the laboratory of Professor Emmanuel van Auberen at the School of Medicine in Nice, where he studied the role of microRNAs in the regulation of insulin signaling in pancreatic beta cells. And after completing his doctoral studies, he moved to Boston in 2010 and joined the laboratory of Professor Rohit Kulkarni in Joslin for his postdoctoral training. And during the fellowship, he had major interest in the role of organ crosstalk in adaptive islet beta cell regeneration and pre-diabetic states. In March 2017, he became an assistant professor in the Department of Medicine, Division of Endocrinology, Metabolism, and Nutrition at Rutgers Robert Wood Johnson Medical School, where he studied the role of sensory neuron islet intercommunication in glucose metabolism with an emphasis on the identification of molecular and cellular mechanisms promoting islet beta cell function. And then in December 2022, he was appointed associate professor in the Department of Cell Biology, Anatomy, and Pharmacology at New York Medical College, where he continues to elucidate the role of peripheral sensory neurons in the islet beta cell activity and energy homeostasis. The title of his talk is Sensory-Islet Crosstalk and Glucose Homeostasis. Thank you so much, Abdel. We really are excited to talk a bit more about your recent paper and your current work.
Thank you, Monica. Thank you for the very nice introduction and for having me to speak and talk a little bit about our work. I was saying, Monica, the first time we met was about two years ago at an ADA meeting, both of us looking at the same poster on the vagus nerve and pancreatic beta cell. And fast forward 3 years afterwards, now we're talking about the same thing again at this meeting. Thank you again. Thank you.
That's great.
Thank you very much. So I'm going to just forward this. Okay. So historically speaking, this has started many decades ago, thanks to Claude Bernard, the renowned physiologist. He's probably the first one who actually demonstrated that the nervous system is regulating energy balance. A series of experiments he did by just lesioning, just poking around the brain. And he was the one who showed that indeed the brain is very important for the regulation of glucose balance. It was around the same time that Paul Langerhans, who discovered pancreatic islets, also told the same thing. He said something about that these many organs are very, very heavily innervated. I think I do not know how much work has been done around that time in terms of studying the nerves and beta cell, but I feel the discovery of insulin in 1921 has sort of slowed that effort, and things have been sort of picked up a little bit lately because of major advances that we have seen in neuroscience. So I'm very happy to be part of a very small team that looks into the neurobiology of pancreatic beta cells. And so when we started this a few years ago, I just sketched this schematic to show the pancreas and the different nerve input. And you can see in the schematic, for example, they receive the input from the vagus nerve, both efferent pathways emanating from the dorsal motor nucleus of the vagus, but also sensory neurons, which actually constitute about 80% of the vagus nerve that are located in terms of the cell body in the nodus ganglia. From the sympathetic nervous system perspective, they receive spinal nerves from, in terms of sensory afferent innervation from the dorsal root ganglia, and the efferent pathways are from the celiac/mesenteric ganglia. And so we have been trying over the last few years to manipulate this in the most specific way we can. We started using either drugs or surgical ways, but I think now we're moving forward and being able to use genetics to manipulate afferent pathways of the pancreas. And so again, when I started doing this almost 5-6 years ago now, we started sort of doing some imaging. We used some of the cool technique that neuroscientists came up with. And here we have used clarity. We modified a little bit to adapt it to the pancreas, but it's actually really no different. We just formed this sort of hydrogel mesh around the pancreas to maintain the structural integrity of the tissue, and then you clear all the lipid just by submerging the pancreas in SDL solution to take all the lipid out. By doing so, removal of the lipid allows you to clear the tissue and limit most of the light scattering, but also removal of the lipids allow you to diffuse the tissue with different probes, different antibodies, and fluorophore. And so by doing so, and then by adding an additional layer of clean by submerging the pancreas in a what you would call a refractive index matching solution, you clear the pancreas further, again, to limit light scattering. And so this is an image that sort of looks at the brain here, how we're able to clear the brain at a time within just 7 days. And for the pancreas was actually a lot shorter. We can clear the entire pancreas within only 3 days. And so we're able to section very large chunk of tissues. I'm showing the first one here is about 5 or 6 millimeter large, stained with tyrosine hydroxylase as a marker of the sympathetic nervous system and glucagon as a marker of endocrine cells. And you can see how much innervation actually there is in the pancreas. This is a zoom in of the same image here. Again, you see Th positive neurons coming in along the blood vessels and projecting within the islets. We have used a variety of different markers, including beta-thromboxane, GFAP, and so on. You can see there's a really heavy innervation of the pancreas. These are a couple of videos probably have showed in the past, again showing the same image as earlier, where you can see a tremendous amount of nerves projecting directly within the islet. And on the right side is another image showing this is a marker of sensory neurons called TRPV1, which I'll be talking about it in the next few slides. These are neurons sensitive to heat, but it turns out they have also important roles in aspects of glucose and lipid homeostasis. And so this turned out to be not a mouse thing in humans as well. The pancreas is known in humans to be also rich in different kind of neurons. And this is a section from, this is a white male, age 76, with a near normal BMI, staying with glucagon, again, a marker of endocrine cells, and PGP9.5 as a pan-neuron marker. And you can see a lot of nerves coming in and project directly inside the pancreatic islets. Now, I want to go back and talk a little bit about TRPV1. We have conducted a series of studies to address the role of TRPV1 neurons in pancreatic beta cell. There has been few studies on this to really document the role of TRPV1 in longevity and lifespan in mice. And some of that has been attributed to improvement in metabolism. And so we thought to see if there is any role of these neurons in pancreatic beta cell biology. And so in these studies, what we did, we used capsaicin as a way to desensitize and denervate the mice from these neurons and see how that would affect glucose homeostasis and then beta cell function. And so the way we did this, we just inject the capsaicin here in the sub-Q, and for over 30 days or so, we have phenotyped these mice. And so what you can see here, that in male mice, we're able to see that capsaicin, or chemical denervation of TRPV1 neurons, was associated with a major improvement in glucose clearance without affecting insulin sensitivity. We did not see any change in body weight or random fed blood glucose, not in random fed insulin levels. The mass, the body weight, and the weight of different tissues was pretty normal between the two. If anything changed here is that insulin secretion was improved in this mice. Now, the surprise we had when we did this experiments that time is that these effects were only observed only in male mice. If we repeat the exact same experiments in females, we did not see any difference there. We took the male mice and did additional experiments to look at a little bit of other aspect of energy balance. We did not observe any differences in their activity, in oxygen consumption, CO2 release. If anything, really, these mice, they tend to eat a little bit more than the mice that were injected with vehicle. And then we learned around that time, there was actually a paper that was published in PLOS Biology by a team at Albert Einstein, showing that there's also a very discrete population of TRPV1 neurons in the hypothalamus that get activated upon exercise, and that limits a food intake. And so it's normal that we now denervate TRPV1 neurons. You see an accumulation of accumulative food intake behavior there. Okay, and so then we wanted to be a little bit more specific, and so we injected now capsaicin directly in the pancreas through a pancreas intraductal injection. It's a small amount that we inject one time, and then we phenotype the mice the same way we did in the systemic injection of capsaicin. And again, we were able to recapitulate the exact same phenotype, and that is to say, in male mice, capsaicin treatment has improved glucose clearance without any change in insulin sensitivity. And again, this change in glucose clearance was associated with enhancement in insulin secretion upon glucose stimulation. We did not observe any change in aspects of beta cell mass, beta cell growth. And again, in female mice, there was absolutely no difference between the controls injected with the vehicle and those injected with capsaicin. We added a new model, another model, where now we went surgically and removed the ganglia that we know project in the pancreas. There are few, the ones that project in the pancreas, I think, span between somewhere thoracic, dorsal root ganglia and thoracic 8 or 9 down to lumbar 1 and 2. So we went to pick actually 3. We just harvested 3 of them from T11, T12, and T13 unilaterally. And you can see here that there was really sufficient just through 3 dorsal root ganglia from one side was able to improve glucose clearance in a mice and improve insulin secretion as well. Again, we did not see any change in beta cell proliferation, beta cell area, and beta cell mass. And again, in females, this experiment did not really yield any results. This was a very strictly a male phenotype. And then we asked the other question around, which is, well, does beta cell activity in any way enhance the activity of these sensory neurons? And so we learned how to do dorsal root ganglia culture, and here we actually sort of trypsinize the cells, the ganglia, to obtain a population of cells that we cultured, and then we incubated with those calcium indicators and looked at the effect of insulin on sensory neuron activity. And so here in this short video, you can see that upon adding insulin, you can see a very nice activation of calcium influx within the cell. And you can actually quantify this. You can see that within control, you do not see much of a change. But when you add insulin within the next 2 minutes or so, you see a very nice increase in calcium influx indicative of neuron activity. You can also take the culture media from these experiments and measure CGRP. Calcitonin gene-related protein is it's a major neuropeptide released by sensory neurons. You can see there's a trend to be increased in these experiments. You can also take obese mice and look at CGRP within the serum. You can see there's an increase in CGRP there as well. And so we conclude from these studies that upon beta cell activation by glucose insulin release will activate DRG. And then DRG now, once it's activated, it will release molecule, CGRP, and potentially other molecule that would now shut off pancreatic beta cell. And that's why basically when we add capsaicin or remove this DRG surgically, you virtually see an increase in insulin secretion. Okay, and so now I'm going to talk a little bit about why do we see these differences, the sex difference, why this is more dominant in males than in females. To do this, we used viruses, trophic viruses for neurons. This pseudorabies viruses, they infect only neurons. So when you put the pancreas, they would infect terminal ending and ascend to the cell bodies. And so in these experiments, we use 2 kinds of viruses, the PRV that express a GFP protein that was injected in the pancreas, and we used another PRV with a red fluorescent protein that we injected in the liver. So after a few days, we harvested dorsal root ganglia and also nodose ganglia. So we sectioned those and look at a little bit the fluorescence, and by counting the amount of fluorescence in each of the cell body, you would have an idea about the density innervation in the pancreas and the liver. And so I'll start with the dorsal root ganglia here on the right side. You can see just by looking at the image that the amount of positive cells for GFP in females is lower than in male. This is, if you look at now RFP, there has been no difference. The amount of neurons that go to the liver is pretty much the same in males and females. We were surprised to see that some of the neurons, they express both GFP and RFP. And so you basically have dorsal root ganglia neurons that would bifurcate and have one branch in the pancreas and another in the liver. This trend here was actually quantified in these images around here. So you can see the amount of sensory neurons in the female pancreas is almost halved compared to the male pancreas. This is true in the left notus ganglia, the right notus ganglia, and you put them together, you see the exact same thing. Now, if you look at, sorry, this is the notus ganglia. I was actually referring to the DRG here. It's halved in here. No difference in the liver projecting neurons. And the same trend, as I mentioned, was also observed in the notus ganglia, sensory neurons of the vagus nerve. So this indicates that somehow this difference in males and females somehow has some neurodevelopmental origin, where the amount of neurons it's not the same in the two sexes. We have repeated the exact same experiments. Now we just superimposed a new N as a pan-neuronal marker and counted specifically those cells that are positive for new N, showing this actually neuronal cells that were labeled. The second hypothesis we want to address is whether sex hormones are playing a role in this sex difference. And so to do so, the first thing we did is we just quantified the amount of terminal ending of these CGRP positive neurons in pancreatic islets within the pancreas between the difference between males and females. So this is very complementary to the PRV experiment. It's just that we're not putting any virus here. We're just looking directly at the pancreas and see if there is more CGRP nerves than in males versus females. And it turns out that it's true. There is actually more terminal ending surrounding islets in the male pancreas than in female pancreas. Now, if you do orchidectomy, if you remove testes in males, you will end up with an amount of CGRP innervation in the peri-islets that is very comparable to the one that you would see in females. So it's likely that testosterone is actually influencing this amount of nerves that are projecting in the pancreas. And so based on this, now we went back and repeated all this experiment that I showed you earlier and superimposed gonadectomy experiments. We did both in males and females, orchidectomy and ovarectomy. Again, as I mentioned earlier, the treatment with the capsaicin will improve glucose clearance secondary to improvement in insulin secretion, not insulin sensitivity. However, when now we do orchidectomy, you can see that this effect is completely lost. There is no difference now in glucose clearance, not in insulin secretion. And when you do this now in females, ovarectomy did not change anything at all. The female mice were not sensitive to capsaicin, and they are still not sensitive to capsaicin after ovarectomy. So this clearly shows that testosterone is actually a male, the male hormone is the one that is guiding the sex difference in terms of the amount of nerves, structurally speaking, but also at the functional level. The last experiment we did, which is sort of the hypothesis that these sex differences can be driven by factors that are completely independent of sex hormones. And the way we did this, we co-cultured pancreatic islets with neurons from the DRG, and we're able to do both same sex co-culture or opposite sex co-culture. So here in the left side here, the upper left side, you see male islets with male neurons. As you would expect, when you treat islets with glucose, you have an insulin secretion here is increased. When now these islets are in presence of DRG neurons, you see a very higher basal insulin secretion, but GSIS itself is completely blunted. And this is typical of insulin resistance. Most of people in mice that are insulin resistant, they usually have very high levels of insulin, but then the ability to put out more insulin response to glucose is somewhat limited. Now, this was observed again only in the male-male combination. When you do female islets with female neurons, you do not see the difference. First thing you notice that the female islets will release more insulin in response to glucose than males. They have a very superior GSIS compared to male, but then neurons do not have at all affect their ability to release insulin, neither in the basal condition nor in the stimulated condition. Now, we do not know at this level whether this is dictated by islets or dictated by neurons. To know the difference here, we have conducted, as I mentioned, opposite sex co-culture experiments. So for example, here you would see pancreatic islets co-cultured with DRG neurons from females. And you would see that the inhibitory action of DRG neurons that we have seen here in the beginning is completely lost when we culture male islets with female neurons. And the same thing here now, when you're going to use female islets, the male neurons have absolutely no inhibitory effect on pancreatic islets. So we conclude from this experiment probably there are factors released only by male neurons that inhibit insulin secretion, and somehow that female islets are resistant to this inhibitory action. We went on and tried to determine the factors that are different between males and females, the ones that are released in males but not by females. And so to do so, we conducted these experiments where we took DRG neurons from very young mice, our 3-week-old mice, sexually immature yet to limit any action of sex hormones here. And so we isolated this DRG, we cultured the neurons, and after 3 days, we stimulated them with a KCL to enhance exocytosis. We took the culture media and did a proteomics to do secretome analysis and used the cells for RNA. We harvest RNA for RNA sequencing. And so here we're looking at RNA-seq analysis, looking at male versus female DRG neurons. As you can see very clearly, they separate very, very nicely. And again, these are 3-week-old mice. They were not subject to any sex hormone yet at this stage, except that maybe the neonatal testosterone surge occurred on day 1. But otherwise, they have not been exposed yet to what you would call activational sex hormone. And so at this stage, you can see that you have very nice separation between transcriptome of male DRG neurons versus female DRG neurons. There are a few genes that go up in one compared to the other. And you can see here we have taken about 15 genes or so, and we try to validate them by RT-PCR. And you can see there's a very nice correlation between the RNA-seq and the RT data. And again, this is just to show that this is very reproducible among different replicates here. The major differences really that we saw here is that most of the genes that seems to be regulated in the male DRG neurons are things that are related with cell cycle, proliferation, and things like that. In the female neurons, we were able to see a lot of signaling pathway in the insulin cascade that was more enhanced in these female neurons compared to the male ones. And then we looked at, as I mentioned, secretome analysis. Again, things here separate also very, very nicely. We're able to identify molecules that seems to be enhanced in males versus females. And we are currently looking at these molecules in the context of insulin resistance and identify those that they may be implicated in aspects of growth and function in this particular setting. Okay, so I want to move now to another chapter, which is look at the role of the vagus nerve in beta cell growth and function. I think we're all familiar with the idea that during the setting of insulin resistance, beta cells, they compensate by increasing in mass and function, and that with time, if this is not corrected and glucose levels is not maintained in normal range, beta cells ultimately die. My lab has been for a while now interested in understanding how beta cells compensate to insulin resistance, and the possibility of identifying factors allow beta cell to compensate properly under this metabolic insults. I want to briefly discuss this paper. This is truly the paper that actually got me into studying neurobiology of beta cells. I was not trained as a neuroscientist, trained as a beta cell biologist, but I saw this paper when I was a postdoctoral fellow that described the role of the vagus nerve in adaptive beta cell growth and function. This was published almost 15 plus years ago now. What basically this team actually did, looked at the effect of capsaicin, again, the same drug that desensitizes sensory activity in the context of insulin resistance and beta cell growth. As you can see here, this is again the typical results you would obtain when you, in mice with insulin resistance, showing, for example, here, beta cell growth is increased in normal conditions without capsaicin, and pancreatic insulin content is increased as well. However, now, if we desensitize the vagus through injection of capsaicin, you can see that these both effects are completely abrogated. And they went on and actually did pancreatic vagotomy And again, you will see that in the contrary to in contrast to control condition, cutting the vagus nerve branch that goes to the pancreas completely blunt this effect at both the function, but also the growth as well. And this as a control, if you do hepatic vagotomy, you do not see these effects. And so from here, we get this idea that, well, the vagus nerve must be releasing something that is helping the beta cell to compensate to insulin resistance. And so to address this question, we used this mouse model, Is961 model, which is most of us now are familiar with. Is961 is an antagonist of insulin receptor. When infused in the mouse, you create a chemical state of insulin resistance. And you can see within really few days, day 3 here, the mice become hyperglycemic, severely glucose intolerant, severely insulin resistant, the dramatic increase in beta cell mass and beta cell function by having more insulin. And so we thought to interrogate the repertoire of the vagal sensory neurons transcriptome and see if you can identify factors that allow this compensation to occur. And so the way we did this, we injected 6-week-old mice with the compound and for over 2 weeks. And we harvested the ganglia, we extracted the RNA and then we did an RNA sequencing. And we end up looking at this gene called FGF3, a member of the AGF families, about 20 plus members, potentially interacting with 4 different receptors. We end up looking at FGF3 because it binds to 2 receptors, FGF receptor 1B and 2B, which are known to be to have genetic variants. Its genetic variants are associated with type 2 diabetes. And so I'll go through this briefly bioinformatic analysis. You can see again that these 2 groups, the control and the Is961, separate very nicely when you do principal component analysis. There are a few genes that you would see going up in one condition compared to the other. This is very reproducible among 3 replicate. Gene ontology analysis revealed increase in pathways that are related to regulation of insulin secretion and beta cell activity. And this was confirmed by doing another set of analysis using gene set enrichment analysis. You'll see the same thing here. And so we're able to confirm that FGF3 is indeed increased in the Is961 model in acute setting, but not when you put this in long-term insulin resistance. And so we're able to get FGF3 and we put that in a virus and we express that in a mouse model, just a wild type mouse, and see if FGF3 has any effect on glucose homeostasis. And so here we use an AAV9 to just inject this in the tail vein, and we phenotype this mice over time. So we're able to confirm indeed that FGF3 is overexpressed in the liver of this mice. No change at this point on body weight, although the mice tend to have a lower glucose levels compared to the GFP injected mice. However, the mice, they have very nice improvement in their glucose clearance. No change in insulin sensitivity, no change in random fed insulin levels. But when we simulate now the mice with glucose, you see a very nice improvement in GSS at both time points here. You can see 5 and 15 minutes. When we started this project initially, I was hoping to get at the bottom of factors that allow beta cell to grow. And as disappointing as this may seem, we did not see any effect actually on beta cell proliferation or growth. We are looking at here at by Ki7, and I believe we have done other markers as well. No change there, no change in eye area and beta cell mass. And looking at RER, you can see that FGF3 have a higher RER, which is indicative of a usage of carbohydrate as insulin levels is higher in these mice. Now, we did another set of experiments where we showed that FGF3 actually acts directly on pancreatic eye to potentiate GSIS. So in these experiments, you can see we cultured eyes in low glucose or high glucose. And with FGF3, you can see that FGF3 indeed potentiate glucose-stimulated insulin secretion. And this action now, as you can see, is inhibited when we add small molecules that inhibit FGF receptor signaling. So this indicates again that FGF3 acts directly on beta cell to stimulate insulin secretion in FGF receptor dependent manner. Now, the next set of experiments really deals with adding FGF3 in the notus ganglia, where we found it upregulated, and see if that would affect glucose homeostasis. As a sort of a mock experiment, as we train ourselves to do this, we're able to now to open a mouse and look at the nervous ganglia and be able to inject a virus in. And so in this experiment, for example, we're able to put an AAV9 expressing crude recombinase in AI14 mouse. The AI14 mouse is the TD tomato CRE-dependent mouse. So when you inject the CRE in, you can see that after a few weeks, you have a very nice stain of TD tomato within the nervous ganglia. I should say at this point that the nervous ganglia in the mouse is fused with the jugular ganglia, which is very different from human. So sometimes you can set a little bit of signal up there as well if you need to get a little bit farther than the nervous ganglia. But usually we detect most of the signal actually in the nervous ganglia because it had a very nice bumpy shape as opposed to the jugular ganglia, which is very, very amorphous here. Okay, so now that we become a little bit familiar in doing this, we're able to do the experiment of injecting FGF3 directly in the nervous ganglia and see what happened. And so again, it's experiments on young mice, AAVs overexpressing GFP or FGF3 injected directly in the nervous ganglia. And you can see again, we didn't see any change here in blood glucose and body weight, but GGT is largely improved in the condition where FGF3 now is overexpressed in the nervous ganglia. This seems to be secondary to enhancement of beta selectivity, not insulin sensitivity. ITT did not reveal any difference in FGF3 mice. Oxygen consumption, CO2 release were improved. We did not see change in RER. And surprisingly, in this experiment, we also have seen effects on energy expenditure and physical activity. And we think the reason why is because injection of FGF3 in the nervous ganglia will not only go to the periphery, but obviously will go to the brainstem where you can have some central effect there as well. And so precisely in order for us to try to be as, and there are obviously other organs that are innervated by the vagus that also could have an effect. So we're trying to limit as much as we can the expression of GF3 to only vagal sensory afferent. We used AV retrograde to overexpress now FGF3 specifically in those neurons. And again, this is again one of those mock experiments where you can see a mouse. This is a pancreatic intraductal injection of GFP using an AV2 retrograde. So by putting this in the pancreas, again, it will affect neurons in the pancreas and it will ascend to nervous ganglia and express your transgene or your gene of interest. Again, in this case, it's GFP. This is the left nervous ganglia on the top, the right nervous ganglia in the bottom, stained with PGP9.5. And again, you can see you have a very nice signal of GFP and you're getting retrograde transfer of our insert. Here, I want to say that we were able to see that there are about 5 to 10% of vagal afferent that seem to be innervated in the pancreas. The difference may be between 5 and 10 probably is depending on the injection itself, but we were not able to go really beyond 10%. And I think this is pretty consistent with some of the data they have seen out there on people doing the same thing. Again, controls is again the same experiments. We're just showing that in the pancreas, we do not see GFP within the islets nor within the pili islets. So basically, we just infect neurons that are projected in the pancreas. And so here I'm showing two experiments, one gain of function and one loss of function of FGF3. We have overexpressed FGF3 now in pancreatic vagal afferent using AV retrograde. And again, you can see in this case that we have improved glucose clearance secondary to an increase in insulin secretion without any effect on insulin sensitivity. And again, you can see here these effects on energy expenditure and activity, now they are actually reduced. We do not see that difference between FGF3 and the control mice. And in the loss of function experiment now, we have injected AV retrograde expressing CRE in a mouse model that has FGF3 allele flux. In chow diet conditions, we did not observe any effect on glucose clearance nor on insulin secretion. However, when you put mice on high-fat diet, you can see that the mice now, they have altered glucose clearance that is secondary here, as you can see here, to this very large blunting of insulin secretion in response to glucose. Okay, and so the last experiment that I want to tell you about, and this, for me at least, it was sort of the most informative. Overall, we have been doing this now for almost 2 years actually on this project. We have been really operating under the assumption that FGF3 somehow is released from afferent neurons and affect insulin secretion directly by interacting with the beta cell. And that may be true because we have added FGF3 on the beta cell and it does so. So the possibility we cannot negate it. However, the experiment here actually is telling us something very, very different. And so this experiment was done in collaboration with Dr. Louis Olua at Duke University. What he did here was to cut the vagus and stimulate, this is an electrical stimulation of the proximal tip of the vagus. And so basically here what we did, we stimulated the afferent pathway, but there is no efferent pathway that is active. And yet, when we do so, we still see that the blood glucose is largely improved after a bolus of glucose. GTD is largely improved as probably a result of increase in insulin secretion. So this was a subdiaphragmatic vagotomy, and he even actually went on and did a cervical vagotomy. And this is very difficult experiment because when you do this, the mice, they die within the next 24 hours or so. So he was actually very able to do tracheostomy and keep these mice alive and do the experiment. Again, this is very, very difficult because you have absolutely no connection now to any viscera. And yet, when you do these experiments, again, you see that the afferent stimulation alone was without any efferent pathways. You see that it was able to improve glucose clearance and enhance insulin secretion. This, at least for me, it does two things: that the idea of vagal reflex might be a thing, but it's not the only thing. It's possible that when you activate the afferent pathway, you have a vagospinal reflex that actually puts in place, or even molecules released by different regions of the brain that can travel systemically and affect beta cells. And so we're very happy to see the story actually published in Development Cell only a couple of months ago. And now I want to tell you a little bit about what we're trying to do moving forward. At this point, we become very, very obsessed about the idea of identifying the very cell subtypes that go to the pancreas, what are they made of, and what do they release. And so we're doing this in two different ways. We're using this mouse model called RiboTag, in which you can tag a ribosome and purify RNA and sequence your cells of interest. And the way you do this, again, you can inject just this A retrograde in the pancreas, you harvest the neurous ganglia, you IP the ribosome and the RNA, and you just sequence and you see what you get. The second model we're using, again, is AI14, where you can specifically label the cells of interest. And again, we inject the virus in the pancreas to label cells with T-tomato. We just take them out, we dissociate them, and we do a single-cell RNA-seq. We think that by doing these experiments both together, we're going to be able to get the markers and the identity of those cells and manipulate them in a way to affect beta cells. And I have one final slide just to use some very preliminary data. We're able to tag and label pancreatic beta cell, not pancreatic, I would say pancreatic afferent. It's very hard at this moment to do pancreatic beta cell afferent, but we're able to do that for pancreas afferent, label the cells, dissociate them, and do a single cell RNA-seq. I will not go through all this, just to say that we have identified about 28 clusters, just like a lot of other researchers have did. All these are neurons because they express characteristic markers of neurons. Most of them, they have a FOX2B gene, which is very characteristic of neurous ganglia. And also some of them, they are PRDM12, which is characteristic of jugular ganglia. I was very happy to see indeed that there are two populations of cells that express T-tomato and the Cre. Cre is the one we injected ourselves, and T-tomato is a result of the recombination. So two of the cells, based on this very initial bioinformatic and assessment, there are two kind of cells projected to the pancreas. And so the question now, of course, we're asking is, well, what are the specific molecular markers of these cells? What do they respond to? What do they release? How do they affect beta cell versus alpha cell? How do they behave in insulin resistance and diabetes, for example? These are all the questions that we are very much interested in investigating in the next 5-10 years, hopefully. And so with that, I hope I'm still on time. I will just stop here for question. I want to thank, of course, people in my lab, both the past members and the current members. Many thanks to all our collaborators and previous and current funding. So with that, I'll stop here, and I'll be taking any questions. Thank you.
Thank you so much. That was fascinating. Abdel, I'm opening up for questions. You can drop something in the chat. can see here, or you can unmute yourself and ask a question directly.
I can start.
Thank you, Medina. How are you?
Hello, Abdel. Very, very nice presentation. Thank you very
Oh, hi,
much.
Medina. So good to see you in person.
Good to see
How are
you
you
as well.
doing?
Good, good.
Good.
I haven't seen this paper yet, so I'm looking forward to reading it, the FGF paper that just came out. I was wondering what was the endogenous level of FGF in those vagal neurons, and what is the normal FGF3 level in the plasma? Because you're overexpressing FGF in those neurons to see those phenotypes, metabolic responses. But what is it endogenously?
Right. So I do not have the sort of, we know it's expressed, so it's certainly detectable, but it's largely increased in the setting of acute insulin resistance. The fold change by RT-PCR is about 3 to 4 folds. Um, um, and again, that was observed only in acute settings. We're not able to see that if you put mice on half of that for a very long time. There is some increase, but it's not as dramatic as in acute settings. Now in the plasma, we did not look in the plasma because there is no good, I would say, ELISA assay allow you to look at FGF3 in the blood. I have not really seen any good case to do that. That was one of the controls we actually wanted to do. I think in the experiments when we expressed FGF3 to AV9 in the liver, I'm expecting that there will be some FGF3 in the circulation, as it should be. But we really wanted to restrict that expression now only to nervous ganglia first, and then in the second set of experiments only to pancreatic vagal afferent. And even in those settings, we're able to see actually very robust data, as opposed to if you look at just the AV retrograde experiments, it has only data that are positive on beta cell and GTT with absolutely no effect, for example, on physical activity and energy expenditure. Those were observed when we put FGF3 in the nervous ganglia, because we think that would affect directly the branch that goes to the brainstem. As I think more about this, and a lot of viewers ask me this, a better way of doing it is to use what we call a dual viral strategy, which is to inject the AV retrograde in the pancreas that is somehow Cre-dependent or flip-dependent, and then go inject the other virus in the nerve gland. That way, you really restrict your FGF3 or any transgene to the vagal-islet axis. Those experiments turned out to be very difficult to achieve, because if you think about it, there is only 5 to 10%, if you are lucky to actually obtain those 10% that go to the pancreas. If you now inject a virus, you are not sure to actually able to get that amount, because injection doses they are very difficult. You are not sure to actually be able to target that 5-10%. And so we had a lot of going back and forth with reviewers on how to do this. And for now, we are not able to do it. The hope that I have from all the sequencing experiments we are doing is to be able to come up with something that is very unique in the nervous ganglia that projects to the pancreas. That way we can use those drivers to directly drive their activity and expression of other transgenes. But as of now, I personally have been doing this only for a few years now. I was not able to come up with anything more specific to do. Does that help a little bit answer the question?
Yeah. I was just really surprised that FGF would be found in neurons, and it has such a robust effect. And if I may ask a follow-up question related to that.
Yeah, of course.
So do you have any data on showing whether this FGF is being secreted from the neurons, and whether it is being secreted in the periphery or in the brain?
We haven't done this. We just actually started looking at culturing nervous ganglia. It's certainly something we'll be looking into. Great question.
Yeah, it is strange, because FGF3 is, related to cell growth and division, like embryonic development, wound healing, bone growth. And it's like, there's some mutations, right, that result in cancers, but is there any other data showing it showing up in neurons?
Right. So there is a phenotype. I'm glad you mentioned cancer, because as we do this, if you leave the mice for up to 10 months, the mice, they will develop a cancer phenotype. But here is another trick to it, is that you can retain all the metabolic benefits of FGF3 without having cancer if you express FGF3 through the route of AAV retrograde.
Mm.
Same experiments, if you do systemically FGF3 or in the nodus ganglia, you would have those benefits in the beginning, and then afterwards, you're going to have a hepatocarcinoma. However, if you now use the AAV retrograde to express FGF3, you have again those metabolic benefits without liver cancer. And so we have gathered a lot of actually of these tissues over the last 2 years. We're going to look into sequencing different tissues and see what happens there in terms of signaling pathways. But I agree with you. FGF3 has been described in development, has a lot of sort of trophic activities in different tissues. It's described in terms of its receptor that is mutated in different types of cancer. But again, there is a way to overexpress FGF3 to again have those metabolic benefits on insulin secretion without affecting liver function or liver proliferation.
I had a quick question for you, Abdel.
Yes.
With the there's a paper with C et al. In 2008 that basically showed there was a diabetic autoimmune targeting, this is in type 1 diabetes, of glia surrounding the islets, right? That contributes to early diabetic inflammation. How might you think about that in context of your work, the glia, that might be interfacing with the ANS innervation?
I think I have seen. I think there was a paper that describes the role. Is it the one on TRPV1? I think it was in Nature Medicine or in Cell.
Yeah, I think that's it. It was embedded in the January 2024 review by Pia Yang and from Denmark.
Yeah, I don't know. I think there has not been a lot of. I have seen that story. It was a very, very interesting story. I have not seen a follow-up later, as sort of as an explanation of how inhibition or silencing of these neurons would basically relieve autoimmunity in type 1 diabetes. It was the only paper that I have seen that actually says that.
Of the glia. Yeah, no, it was
Glia.
like a pathology, notation almost.
Yeah, but it's interesting because it tells us somehow there is maybe no benefit of having a sustained activation of this afferent pathways. This always remind me of this naked mole rat, which is able to age until the age of 30 years old, and it's known to have no sensory. It doesn't have any CGRP, I believe, no sensory neurons. I don't know. We're trying to do experiments where we increase the activity of afferent neurons in a chronic manner and see what happens over time. And I think this probably will yield some result, hopefully.
Yeah, it was really interesting, very meticulous set of protocols, and it's definitely looks like it's a challenging set of protocols as you do these surgeries and everything, but it looks like you're pulling out some interesting data.
It is, yeah. I think the major issue that I found with I'm still happy though that I'm doing this, is that you cannot rely much on genetics to do this, because as I mentioned multiple times, when you use CRE drivers to manipulate endocrine cells, you somehow end up manipulate things in the nervous system as well. I think we've gotten better with the insulin promoter, where we have now drivers that are more specific to beta cells that do not leak in the brain.
Yeah.
And for other hormones, I'm not sure if we've gotten there yet. We're talking earlier about glucagon and GLP-1. There's a lot of GLP-1 in the brain, and that does a lot of things. I know. So we're trying to do surgeries, injecting in different tissues. But I think if we sequence, and this is not very easy also, to obtain markers of specific population of neurons they can manipulate in the most specific way. But I think by doing this together, doing RNA-seq, single-cell RNA-seq, some proteomics data, hopefully we can get some markers to manipulate in the most specific, desirable way, I would say.
Yeah. It sounds like you're on the right path.
Exactly.
So we're at the top, almost at the top of the hour here. I see you're hiring. We'll put a shout out on our platform for you. I'll communicate with you after, and you know, congratulations on this really unique approach. It's really exciting, and we'll keep an eye on your next papers.
Thank you, Monica. Thank you all for
Thanks.
attending. Thank you.
Thank you, everyone. Have a great rest of your day. Bye-bye.
Bye.